By the end of this video, hopefully we understand the key learning outcomes, which are to explain how lymphocytes can recognize specific pathogens, name the lymphocyte types which produce antibodies, explain how antibodies produced by lymphocytes can cause allergies, explain how antigen binding leads to the increase in lymphocyte production, describe the structure of an antibody, and describe the formation of an antibody-antigen complex. This all sounds very complicated, but it's actually very straightforward. So we have cells in our body called lymphocytes. Lymphocytes are the white blood cells of the specific immune response. On the outside of these white blood cells are membrane receptors. Each lymphocyte has membrane receptors for only one antigen protein. When we talk about antigens, it almost makes it sound like pathogens are deliberately putting proteins on the outsides of their cells that the immune system can attack. But that's not the case. Pathogens have normal proteins like every other organism on the outside of them. Because a lot of pathogens are single-celled, they might have a protein that allows them to move towards light sources, to move towards nutrition. They might have structural proteins that allow their cell walls or cell membranes to remain kind of strong, and they might have transport proteins as well. There's lots of different reasons that the outside membrane of a pathogen might show these proteins. It's just that our body can recognize these proteins. And if we take, for example, the protein that allows this pathogen to move toward light sources, we have a lymphocyte type in our body which has a membrane receptor which can recognize that. This lymphocyte is specific for only that one protein. Now, what is good about this is that pathogens actually quite often have many different antigen molecules on the outsides of the membranes. If we just take this one for just now, we have lymphocytes in our body which can recognize all manner of different proteins. But there's no point in having hundreds and hundreds of each lymphocyte type going around the bloodstream at any one time. That's a massive waste of energy, of amino acids, of sugars, of resources. You only really want a lot of the lymphocytes that are needed for that one moment when there's a pathogen that is complementary to that lymphocyte in the body. So what we have is a process called clonal selection. In clonal selection, if a small number of lymphocytes are going around the body, if lymphocyte B recognizes an antigen on the outside of a pathogen that it is complementary for, it clones itself through replication and repeated division, and it makes many hundred copies of itself. So only the lymphocyte types that are actually needed are then replicated through clonal selection. So again, this stops us producing lots and lots of lymphocytes of a type that we don't need at that moment. And you can see an example here. If all of these are different types of lymphocyte, only the lymphocyte that is complementary for the pathogen that's in the body at that one time, which has antigen X on it, will go through clonal selection. There are two different types of lymphocyte that we talk about in the course. We talk about B and T lymphocytes. They are very similar kind of starting functions, but they help to destroy pathogens in very different ways. A B lymphocyte produces a Y-shaped protein that you'll have heard of called an antibody. These, again, just like the lymphocyte itself, have a receptor binding site. So, as we talked about before, the lymphocyte its membrane receptor will see an antigen that it is complementary for, and it will release these antibodies, which again have a binding site. What tends to happen next is that the antibodies will become bound to the antigens. This will inactivate the pathogens. So if there's a pathogen here, it's designed through evolution to be quite small and snaky and kind of move around the body quickly and avoid the immune response. If the antibodies attach onto its antigen, but also attach onto the antigen of a pathogen beside it, and another one, it makes these massive big complexes, and it traps the pathogen into this antibody-antigen complex. The bacterium in this case can't get away, it's not small enough to enter cells, and it can't avoid the non-specific immune response that we talked about before. So these antibody-antigen complexes can then be phagocytosed by phagocytes that we talked about in the previous video. So the antibody-antigen complex stops the bacterium being able to avoid the immune response. Pathogens that try and get into cells are now too big to get into cells because they're in this big complex, and it's easier to notice them for the immune system and destroy them through phagocytosis. T lymphocytes, as I said, start off with the same first step. They recognize the pathogen, antigen, and the membrane receptors. So what you might have thought about by this point is that how do the lymphocytes know that they're not just attacking the normal body cells? We talked about the fact that these antigens on the outside of the pathogen cell, they have a function. They might be structural, they might be sensory, so they might tell the pathogen where the light or the nutrition or other pathogen cells are for reproduction. But we, as you can see here, we have proteins on the outside of our cell. And the body is actually very clever in not allowing any lymphocytes that would be complementary for our body cells. It doesn't allow them to be created, and if they are created, it destroys them pretty quickly. So the body is quite good at noticing the difference between self and non-self antigens. We'll talk about later, occasionally in some medical conditions, the body makes a mistake and will produce lymphocytes that attack its own body cells. In that occasion, you would get autoimmune conditions. So we've had the first step, the lymphocyte has used its membrane receptor to recognize a pathogen's antigen. In the second stage, while still attached at this section here, it will produce proteins. These proteins move into the infected cell. So it's not always the case that the lymphocyte will attach onto the pathogen itself. Sometimes what will happen is that a viral cell, a viral pathogen in particular, will get inside one of our normal body cells. And it will do this to hijack the body's normal processes. It'll come in, it'll go to your nucleus, and it will force your body's normal cells to produce more pathogen, more viral proteins. Your body cell will then put them together for the pathogen, and then when the cell is completely full of these viral invaders, it'll die, it'll explode, and the pathogens can then rush into the bloodstream, quickly get into another body cell, and the cycle goes round again and again and again. Luckily for us, when these viruses go into the body cell, they quite often leave little port proteins on the outside of the membrane. So actually, the normal body cell now has a viral protein on the outside of it, and that can be recognized by our lymphocytes. So we've recognized that protein on the outside of an infected cell, we've attached onto it, and we've released a protein which leaves the lymphocyte cell and goes into the infected cell. What that does is it encourages the infected cell itself to produce self-destructive enzymes. So these enzymes will destroy the inside of this infected cell, will completely kill itself in order to get rid of this viral invader. So all the nucleus, all the mitochondria, everything is digested by these enzymes, and that includes the viral protein as well. So we've recognized the cell has been infected, we've released proteins that go inside the cell, and these proteins, these transcription factors, will tell the infected cell to destroy itself. That's a really, really clever process. Unfortunately, as I said before, that can make little mistakes. Things like type 1 diabetes quite often are autoimmune conditions. And what that means is that the body produces T cells, T lymphocytes, that will attack your normal body cells. In the case of diabetes, it attacks the cells that produce insulin in the pancreas, these beta cells. And that means that no matter how many times the body tries to repair these cells, lymphocytes will keep coming back and driving this immune response. Things like Crohn's disease, things like certain types of arthritis. There's lots of autoimmune conditions, auto meaning self, immune meaning immune system. So you are using your immune system against yourself. In autoimmune conditions, it's the T lymphocytes that do this. The B lymphocytes don't get away easy either. They are responsible and at fault for allergy. So we talked about the B lymphocytes. They recognize pathogens by their proteins using the membrane receptors. They should only recognise pathogens though. If you have something like hay fever, pollen, when it gets into your nose, there's a lot of immune molecules in your mucus, and they recognise the pollen. If you have hay fever, the body gets a bit over enthusiastic and tries to destroy that pollen. And it does that through driving the B lymphocytes to produce antibodies and to attack that pollen. This tells the mast cells that we learned about in key area 5 to produce the inflammatory proteins like histamines, and that brings blood to the area. You get runny nose and red eyes and some nasty hay fevery effects. It could be more serious than this. If you have something like a nut allergy, this response can actually happen in your airways. So you eat nut, some of the nut protein gets into your respiratory pathways, and the B cells produce antibodies. This drives histamine to be produced by the mast cells, and it causes inflammation in your airways. So it can actually mean that not only would you get a swollen and puffy face, but your airways itself would start to become inflamed and would swell. And obviously, if in very serious cases, this could cut off oxygen into your lungs and kill you. So, funnily enough, if B lymphocytes produce histamine, we can take tablets like loratadine and Claritin that would be antihistamine. So these literally just reverse the process of histamine production. So it's quite a clever thing. When you think about allergies, think about antihistamines, and that will help you remember that it's histamine produced by the mast cells that causes this allergic reaction. So memory cells. We have heroically defeated the invader at this point. So we've produced our B and T lymphocytes. We've used our non-specific immune system to destroy it. But what next? Do we want to have to go through that massive kerfuffle of working out what's happening, mounting a response, and slowly building our clonal populations of lymphocytes again? I would say no. So instead, what your body does is some of these lymphocyte cells are able to survive long-term as memory cells. So the first time you come in contact with something, the first exposure, you have quite a slow response as your body works out what is attacking it. It produces, quite often goes through quite a slow non-specific immune section, and then it produces a little bit of lymphocytes to see how serious the infection is, and then a little bit more, a little bit more, a little bit more. So it's quite a slow process. You don't get a lot of lymphocytes produced, and you quite often, because your body's trying to reserve energy and resources, you end up with symptoms because the pathogen is able to divide and replicate to quite a large degree. But these memory cells mean that the next time your body is infected with that invader, it knows exactly what's happening. It's done this before. It can mount a very quick response. It can mount a very quick response. It can mount a response that has a lot of lymphocytes being produced, and it can do it a lot earlier on. This response also tends to last for longer. So you can see the area under this curve is a lot smaller than the area and length under this curve. So it's quicker, you get more lymphocytes, and it lasts longer. And that means that your body completely gets rid of this infection probably before you even become symptomatic. This is one of the key parts of immunity. And at the moment, during the coronavirus outbreak, it's something that people are talking about. In the next key area, we talk about vaccination and the fact that it's nice to produce these memory cells, but first you have to get the infection. But if only there was a way to tell your body that it had this primary infection without actually having to do that. And that's where vaccination comes in. HIV is the final part of this key area. So HIV is quite a nasty virus. It is the human immunodeficiency virus. It attacks and destroys T lymphocyte cells. It actually quite often hides inside the lymphocytes, which makes it very difficult for your body to attack it because it's hiding inside the actual immune cells themselves. As HIV destroys these T lymphocytes, it leads to their depletion over time and eventually can lead to the development of an acquired immune deficiency syndrome, or AIDS. So when the T lymphocytes get to such a low level that they're very ineffective, you have AIDS. So individuals with AIDS have a weakened immune system and are vulnerable to opportunistic infections, especially things like fungal infections that normally our body would just fight away without us noticing them. These can be quite diagnostic in finding out if someone has AIDS or HIV. So to summarise, when a pathogen comes into the body, it has to go through the physical defences, and the chemical secretions also try and get rid of it as well. If it gets into the body, we have the inflammatory response with histamine being produced by the mast cells that's causing vasodilation and permeability of the capillary cells, allowing the phagocytes and clotting factors to get to the site of the infection. The phagocytes will also produce cytokines, which bring more white blood cells, which the specific response comes under, to the area. So if the if the first stage, what's called the innate or non-specific immune response, gets rid of it, great, you don't need to activate lymphocytes. But if not, these cytokines will bring lymphocytes to the site. They will use the membrane receptors to recognize antigens on the outside of the pathogen, and we'll go through clonal selection. So that lymphocyte will say, hey, this is for me, I will make more of myself. So lymphocytes can be B lymphocytes, which produce antibodies to form antibody-antigen complexes and kind of inactivate these. The non-specific immune system can then come back into vogue, and it can use phagocytosis to get rid of them. Or T lymphocytes can attach onto the pathogen molecule and produce proteins that cause that pathogen or infected body cells to kill itself. We've talked about memory cells being lymphocytes that are able to survive past the infection, and they give us this secondary response. In things like HIV, the human immunodeficiency virus, this virus actually attacks and lives inside the lymphocytes, which makes it more difficult to get rid of. If you have HIV for quite a long time, it can develop into an acquired immunodeficiency syndrome, or AIDS, which causes a weakened immune system and makes you more vulnerable to opportunistic infections. So we should have covered all of the points in that box. It's quite a long video, so thanks for staying with me there. If there's any of that that doesn't make any sense, go back and watch the appropriate section, or please do ask your teacher.